Assalamualaikum warahmatullahi wabarakatuh. We are from Group 6 want to present a journal about generation of multiplex mutagenesis population via pool CRISPR-Cas9 in soybean.
The study is about soybean, which is a globally important crop representing a major source of the oil and protein for human diet and feedstock consumption. A legume with modulation of soybean plants also provided environmental benefits to farming practices. Since the announcement of the soybean draft genome, functional characterization of all 46 anti-56,000 annotated genes has been the primary goal in soybean genetic studies. Genetic mutagenesis, including chemical radiation, that is X-rays, gamma rays, and fast neutrons, and transformation induced, which is tDNA and transposon insertion methods routinely used as forward genetic strategies. However, the complexity of the soybean genome, which is paleopolyploid genome, limits the success of these approaches. CRISPR-Cas9 has been successfully adapted in various plants and organisms. Multiplex mutation can be generated by either cloning multiple sgRNAs or targeting homologous regions by a single sgRNA. The application of CRISPR-Cas9 on two flower locus loci, GMFT2A and GMFT5A, has successfully modified the flowering time and aided in the expansion of the regional adaptability of soybeans. Diversity in genetic variation can be generated in plants via pooled transformation of CRISPR-Cas9 libraries. The objectives of the video are: first, the feasibility of pooled CRISPR-Cas9 mutagenesis screening in soybean. Second, optimization of CRISPR-Cas9 tools in soybean and future perspectives. Finally, to identify single or multiplex mutants that covered 102 target genes and demonstrated the robustness of the CRISPR platform.
Methodology: first, plant material and growth condition. Soybean cultivar Huachun seeds was used. For hairy root transformation, plasmid were transformed into the Agrobacterium rhizogen strain. For whole plant transformation, plasmid were transformed into Agrobacterium stromycin strain. Soybean were planted outdoor during summer, while during winter planted in chamber. Plant growth and inoculation were measured after 30 days culture. 3 biological replicates were used for each experiment. Second, construction of PGS201 vector. Binary vector backbone was P-Cambia 1300 vector. HYGR replaced the bar marker by restriction, digestion, and ligation between PSP11 and AAT2. CRISPR-Cas9 expressions were assembled with overlapping PCR and NEB. The endogenous promoter sequence were used PM4, PM8, and GMV6. gRNA scaffold and Cas9 were used to nuclear localization signal. Third, searching for single and multiplex sgRNAs for sgRNA design of target gene. Guide RNA space sequence with NGGPAM based on genomic sequence. It will filter using the criteria sgRNA sequence localized to exon region of target gene. Next, GC content of each sgRNA should in between 25% to 80%. sgRNA target the region near 5' half of the gene. Last, single gene-specific sgRNAs contain one mismatch at 3' end for prompt potential of target, while multiplex sgRNA match with two target genes. For sgRNA cloning, with respect to vector cloning, sgRNA fragments were produced by annealing complementary oligonucleotide. To make annealed product compatible with basal digested vector, additional GGATT nucleotides were added to the 5' end of the forward primer. Next, AAC and A added into the 5' and 3' end of reverse primer. Primer synthesis made together with forward primer, reverse primer, and ACL entries, then annealing. Ligation was using annealed oligomer and basal digested PGS201 vector. After E. coli transformation, Positive clone identified by cloning PCR and single sequencing of extracted plasmid. Last, examination of CRISPR-Cas9-induced mutation in plants. Genomic DNA extraction used longer than 5 cm of hairy root individually and leaf disc. PCR-based screening was performed for verification of sgRNA-mediated editing. To detect of each sgRNA, forward sgRNA-specific primer used along with universal reverse primer for PCR. Gene editing was examined by both PCR and sequencing. After PCR, 300 and 1,000 base pair products were purified for single sequencing. Next were subjected to blast analysis to detect potential mutation.
Okay, for the result, first, design and construction of the sgRNA libraries. A new CRISPR-Cas9 vector, which is the PGES201, was developed for genome editing in soybean. In this vector, BASTA was used as the transformation selection marker, and the vectors was successfully induced gene editing to stable the transgenic plants. In order to generate a full CRISPR-Cas9, 70 CRISPR-Cas9 vectors were cloned and pulled into the 16 sub-libraries. Next is the generation of a multiplex mutagenesis population in soybean. Okay, the identification of the sgRNA in the individual plant was performed via the sgRNA-specific PCR, known as SSP. It showed that all 70 sgRNA could be identified in the TO population, and most of the sgRNA present in more than 3 transgenic lines. Okay, number 3 is the heritability of the tDNA and mutations in the T1 progeny. In this figure, sgRNA inheritance and mutant frequency in TO and T1 progeny of the sublibrary AO2. The y-axis indicates the number of examined T1 plants, while the small blocks indicate the examined plants. Colors like purple, green, blue, and yellow indicate the presence of represented of the sgRNA in the plants, while the gray color indicates the absence of the sgRNA, and the stars indicate the presence of the gene editing in the target loci.
Identifications of double homozygous mutant with increased nodule number. Gene RIC1 and gene RIC2 are parallel genes that encode two nodule-enhanced CLE peptide, which have been proposed to be the root-derived signal in autoregulations or nodulation. Figure A shows the DNA sequence of these two targets. SGRI09 and SGRI010 are designed specifically to target these two genes. No mutation were found in the top 10 potential off-target sites for SGRI09 and SGRI010. Figure B shows the predictor protein structures of these two double mutants. In T2 generation, two double homozygous mutant plants with different types of mutation were identified, which cause friction in the protein sequence. All the sgRNA were secreted in T2 plants and could be considered Cas9 free. Figure C shows the expressions of these two genes in double mutant. Expressions of these two genes was significantly repressed, probably due to the actions of cellular nonsense-mediated mRNA decay machinery. Figure A shows the nodule number at 30 days after inoculation. You can see that the two mutant plants has higher nodule number compared to the wild-type plant. This result indicates that other redundant CLE genes might also be involved in LN signaling. Identifications of triple homozygous mutant with decreased nodule number. In Medicago trachytula, LDN1 genes encode an unknown protein that may be involved in processing LN signal and negative regulations of the number. Figure A shows the DNA sequence of SGR012 targets. Figure B shows the predictor protein structure of these three mutants. These three mutants are soybean homologs of LDN1 genes. SGR012 was designed to simultaneously target these three genes. In sublibrary A03, 10T0 plants were identified to carry SGR012. T1 progeny has two lines that carry missense mutation at all loci, and two independent triple homozygous plants from the A3 line were identified in T2 generation. No other SGRN were detected in T1 plants. Figure C shows the expressions of GN-LDN1 genes in the mutant. The expressions of all three target genes were down-regulated and no off-target editing was observed. Figure E shows the nodule number as 30 days after inoculation. In M. truncatula, LDN1 mutant exhibited a supernodulation phenotype. However, in GM-LDN triple mutant exhibited decreased nodulation together with defective vegetative growth. The shoot and root length and dry weight of triple mutant showed significant reduction in compared with the wild-type plant. These results indicate that GN-LDNS genes are required for normal growth and nodulations in soybean, which is distinct from their M. truncatula homologs.
As we know, many of the crops have complex genome structures, such as polyploidy and paleopolyploidy. We will discuss about the first part of our discussion, which is the feasibility of pooled CRISPR-Cas9 mutagenesis screening in soya beans. So in this experiment, they developed a pooled CRISPR platform for the generations of targeted mutagenized soybean populations. The Basel-mediated one-step cloning system allows easy cloning of sgRNAs. The optimized CRISPR-Cas9 factor and transformations procedure induce a high frequency of mutations. A large scale of CRISPR-Cas9 mutagenesis population will be a great value of crop research. There are two examples of pooled CRISPR-Cas9 were constructed. The first one is in tomato and the second one is in rice. The identity of sgRNAs in individual transgenic lines was characterized by barcode PCR and next-generation sequencing. These technical routes offer significant advantages. The first one is high coverage of genomic loci, the second one is low cost of library constructions, and the third one is high throughput in mutant screening. In gene editing practices, obtaining multiple independent transgenic events at the target locus is necessary. It is to determine whether the observed phenotype is indeed correlated with the targeted mutation, or it caused by off-target editing or spontaneous mutations. Due to the relatively low transformation efficiency of soybean, the generations of genome-scale CRISPR population with multiple allele of target genes is highly impracticable. So they choose to generate patches of mutant collections using small pooled CRISPR libraries, which is 5 to 10 sgRNAs. At such a scale, creating small sublibraries is practical with one pool transformations where multiple alleles per sgRNAs. Regardless of the transformation's throughput, a small scale rather than genome scale CRISPR library is preferred for designing multiplex mutant collections with respect to co-transformations with multiple sgRNAs. Different combinations of CRISPR-Cas9 tDNA that target different genomic loci may be integrated into one transgenic plant. For example, they obtained transgenic plants that harbored sgRNAs targeting both GM1 and GMRIC2, which led to identifications of GMRIC1 and GMRIC2 double mutant.
In this article, the discussion has been discussed is about optimization of CRISPR-Cas9 tool in soybean and future perspective. First is about the expression of Cas9 can give a great impact to gene editing efficiency. When they design sgRNA, it also can give important for efficiency. Routine CRISPR-Cas9 tool specialized only for soya bean and other leguminous plant species. Even though there are already CRISPR-Cas9 technique that has been used, additional optimization is still needed. This is because Cas9, for example, Cas9 driven by embryonic stage specific promoter has shown robust editing ability with rare chimeric mutation in several species. Next, CRISPR-Cas9 target site are selected based on the experimental goal. In here, soya bean that undergo multiple editing can overcome gene duplication. And lastly is the prospect of transgene-free gene editing of soybean. Pasta selection and PCR verification is technique that time consuming and also use labor extensive. But for fluorescent marker, it can reduce the work for transgene-free plant screening.
As conclusion, the study demonstrated that targeted single and multiplex mutation could be produced efficiently. Other than that, the optimized CRISPR-Cas9 platform can be adapted as a screening tool in crops with complex genomes and low transformation efficiency to empower genetic studies and molecular breeding in these soybean species.
That's all from us. Thank you.